Hi, it's Catherine. I wanted to give you a tip about using codon charts and to have an example for us. We're going to talk about polydactyl cats. Poly means many and dactyl means finger. So polydactyl cats have extra fingers. This one has 6 toes on the back foot when there's usually just 4. And this kitty has a thumb right here. You can see it sticking off its hand on that bedspread. So it's got an extra finger. And there's like several different sets of extra fingers that cats can have. But what's cool is it's a hereditary condition and it's coded for an autosomal dominant trait. And we're going to learn what both those mean shortly. But what we want to talk about is how we're going to figure out what amino acids are coded for the DNA in the gene that this trait is associated with. Moving to the next slide. Okay, so here's our practice problem. Polydactyly is associated with the ZRS gene. So I've already got a couple pieces of information. Part of the ZRS gene sequence is as follows: CCC, GGG, TCA, ATT. And then the question that we're going to try and answer, which should feel familiar from your quizzes and your homework, is what is the amino acid sequence coded for above? So there's 2 steps we're going to do here. First, because this is a gene, it's going to be made of DNA. So DNA is in our genes. So forgive my handwriting, I'm using my mouse. And to figure out what kind of amino acids we're going to make from this DNA, we first have to make it into RNA. So the first step is we're going to transcribe into RNA. In A. So if the gene has this sequence, I know that this is in DNA and I want to transcribe it to RNA, and then I'm going to use my codon charts. I also have a different hint here that this is DNA, and I can tell it's DNA because it includes the T. So only DNA has T, RNA is going to use U. So I'm going to transcribe this into RNA. So what pairs with C? G, G, G. What pairs with G? It's going to be C, C, C. What pairs with T? That's going to be A, and then G, and then here's where it gets tricky. So if it was DNA, I would put a T here because A and T pair together, but it's not, it's RNA, and so I'm going to put a U. So next one is U, and then A. So this is my DNA sequence here, DNA, and I just transcribed it to RNA. Cool. So now that I have the RNA from the DNA, that was my gene related to polydactyly, I can use this codon chart over here on the right to figure out the amino acid sequence. We have 2 main types of codon charts. We have the square one and we have the round one. I'm going to show you both. So the first thing we do is we go right to the center of this bullseye area and you find the first nucleic acid that you're looking for. So it's going to be a G. So G, then you look for the second one, G, and then you're going out in your radius. And your third one is G. So the amino acid that we coded for with our first codon is glycine. So I'm going to put glycine below it. I'm going to use a different color so we can see them. I'm going to use blue. So GGG codes for glycine. I'm going to do the same thing with this codon, these 3 letters. Start at the inner circle, C. Then I'm going to go to the next ring, C. Then I'm going to go to the last one, C. So my second codon is coding for proline. So pro. I'm going to give you a second. You're going to do the third one in your head, and I'm going to do it silently with you. So going from the inside out, and hopefully you got serine. So our third is SER, and I'm just abbreviating them so I don't have to use my handwriting the whole time. And then the last one. All right. Our last one's actually a stop codon, which is great because that's going to end our protein. The little ribosome will let go of the mRNA and the protein can fold up and it can be made into extra toe tissue for your cat. So our last codon here coded for stop. By the way, there are 3 stop codons, UAA, UGA, and UAG. Perfect. Let's practice it again with the other type of table. Here we are. There we go. So there's our second table. I'm going to go back to my drawing. So instead of starting in the center and moving out along the wheel, now we're going to look at the labels on the sides. So we have first letter, second letter, and third letter.